Okay, so in section 3, we're going to be talking about capillaries as one of our blood vessels. And these are what we also call exchange vessels. So the diameters are really, really small. They're so small that only a single red blood cell can pass through at a time. And even in some places, our red blood cells or erythrocytes actually have to kind of fold so that they're not their complete diameter, so that they're a little bit smaller in order to flow through these capillaries. Walls are really, really thin, as we can imagine, because these are our smallest blood vessels we have. And like I said, only one cell is going to form the entire circumference. There are pericytes, and these are contractile stem cells that can help create new vessels or scar tissue. They also can help stabilize the capillary wall and help control the capillary permeability, meaning what comes into and out of the capillaries themselves. They pretty much supply almost every cell with the nutrients in blood. Some exceptions are cartilage, because cartilage does not have any blood supply to it at all. It's avascular. If we're talking about our epithelia and our dermal layer of our epidermis, our epithelial layer of our epidermis also does not have a blood supply. Cornea and a lens of our eye. And again, if we're talking about something or a tissue that's a single cell layer thick, that function is usually for exchange of different things, because things can easily cross a single endothelial cell layer. So we're talking about exchange of gases, nutrients, waste, hormones between the blood and the interstitial fluid themselves. So all of our capillary endothelial cells are joined together by tight junctions. And tight junctions, a good analogy for that is like a Ziploc bag. So if we put a bunch of liquid in a Ziploc bag, we sealed it correctly, and we turn it upside down, it's tight. Nothing can get in and out. We do have gaps in between these tight junctions that are called intercellular clefts, and these allow for the passage of fluids and some small solutes. There are 3 types of capillaries. We have continuous capillaries, fenestrated capillaries, and our third one, let me go ahead and show you, is the sinusoid capillary. So just a couple things to point out if we're talking about continuous capillaries. If we look at our continuous capillary, this is going to be found where in areas where skin, muscle, lungs, and our central nervous system. These are the least permeable, but the most common type of capillary that we have. And then they also have those pericytes that are associated with it that can help rebuild the blood vessels and have other functions as well. Fenestrated capillaries have large fenestration, so that's why they're called fenestrated capillaries. And fenestrations are just pores. So if we look right here, here's our fenestrations. We can see that we have these pores or these openings between the endothelial cells. The purpose of these fenestrations or these pores is to help increase permeability. So in areas where we have these, they're allowing for more solids and more things to be exchanged from our blood to the tissues. So areas including where there's active filtration like our kidney, absorption for our small intestines, or even if we're talking about endocrine glands and hormone secretion. And the third type is the sinusoid capillary. Sinusoid capillaries are the most permeable, and because they're so permeable, they're very limited in locations, because most of the time we want to be able to control what comes in and what comes out very specifically. We don't want to allow a huge amount of permeability between our blood and the tissues. So where we're going to find the sinusoid capillaries are really in our liver, bone marrow, spleen, and adrenal medulla. And they have these large intercellular clefts and fenestrations. So that's why they're so permeable. And they don't have a complete basement membrane. Again, we wouldn't want to have a complete layer surrounding it, because we're trying to allow for an increased permeability. Okay, capillary beds. So capillaries, they don't function independently. So instead, they form like a network that's kind of woven, and this woven network is what we refer to as a capillary bed. The flow of blood goes from an arterial to a venual, and when we're going from an arterial to a venual, what they have to pass through is this capillary bed. It's also called microcirculation because these capillary beds have very, very small diameters. The terminal arterial, this is a branch off of the arterial that continues to branch further into about 10 to 20 different capillaries that form the capillary bed. And again, the purpose, anytime we're talking about capillaries that make up the capillary beds, is the exchange of different things. So like gases, nutrients, and waste from the blood to the tissues. And then our capillaries are going to drain into what we call the post-capillary venual. So blood flow through these capillary beds is controlled by the diameter of the terminal arterial. So this is what I was describing earlier. So we have an arterial. The blood flow is going to go from an arterial to a venual, but in order to get from an arterial to a venual, we have to have this capillary bed. And so when we have this interwoven network of capillaries right here, that's the capillary bed itself. So how we control the blood flow to a tissue is going to actually be controlled by the diameter of this terminal arterial. So if we constrict the terminal arterial, which means we decrease the diameter of it, we're not going to allow as much blood flow into the capillary bed and then into our venual. So the tissue would not be receiving as much blood supply. If we dilate the terminal arterial, that's going to allow for increased blood flow into the capillary bed and therefore the tissues. So sometimes, depending on what state our body is in, we want to, if we're in a fight or flight situation, maybe send more of the blood to our skeletal muscle and have less of the blood going to our digestive system. So we would dilate the terminal arterioles that are leading to the capillary beds where our skeletal muscle is and constrict the terminal arterial to the tissues, say to our digestive tract. Local chemical conditions and the arteriolar vasomotor nerve fibers, like I just mentioned, those are going to be what regulate and control the constriction and dilation and therefore the blood flow into the capillary bed. So capillaries found in serous membranes of intestinal mesenteries have two additional features that help form a special arrangement of capillaries. So they have a vascular shunt and a precapillary sphincter. The vascular shunt acts as like a channel, and that's going to help directly connect the arterial to the venual. So when we have a vascular shunt, we can bypass sending all of the blood through the entire capillary bed and just kind of quickly send it from the arterial to the venual. So we can see only the highlighted regions right here. The precapillary sphincter, those are these right here showing them open, and this is showing them closed. So they cuff the smooth muscle that's surrounding the capillary, the true capillary, that branches into what we call, off of what we call the meta-arterial. And this is going to act as a valve that regulates the blood flow into the capillary bed. So what controls its opening or closing of these sphincters is going to be determined by whatever the local chemical conditions are. Because again, it's not innervated, so we can't have any nervous system input.